and thank you to the organizers for this opportunity of introducing our work here today, these works. And yeah, my name is Cristina Santini. I work at Celgene. I'm based in Seville, in Spain, and this work is the result of a multi-site, interdisciplinary collaboration. And this works. So lenalidomide is an immune modulatory agent, and it's approved for the treatment of multiple myeloma, del(5q), myelodysplastic syndrome, and mantle cell lymphoma. Lenalidomide causes a reversible block in neutrophil maturation. And for the non-biologists amongst us, I included this diagram, which shows blood development in humans. So chemotherapy-induced neutropenia, for example, is believed to be due to the effect of these drugs at the progenitor level, whereas lenalidomide-induced neutropenia is believed to be due to the block of maturation. Therefore, an in vitro assay was optimized to detect myeloid maturation blockade in vitro. And our goal in this project was to develop an in silico model of this in vitro assay to explore dosing regimens. And these are the steps that we took in the development of the model. We did the data collection, understanding the biology, visualizing the data, translating the hypothesis into the quantitative model, model fitting, validation, and refinement, which brings us to this model building cycle. So regarding the in vitro assay, the assay starts from CD34 positive bone marrow cells from healthy donors. And the assay has two phases. An expansion phase and a maturation phase. And during the first 14 days of culture, we treat the assay with either DMSO or lenalidomide, and followed by a 7-day washout. And these cells are classified as progenitor cells, stage 1, stage 2, stage 3, and stage 4, which corresponds to the mature neutrophils according to their cell surface markers, CG34, CG33, or CG11B. And what I'm showing here is the experimental data. And what I would like to highlight is the compound effect. So you can see that there is no compound effect at the earlier stages of maturation, whereas there is a concentration-dependent compound effect at the latest stage of maturation. The light blue here being 20 micromolar, the strongest concentration tested. And then after washout, these cells that were blocked in maturation are then allowed to mature. So then we translate these hypotheses into a quantitative model. And the schema of our model is shown here. These different compartments represent the cells at these different stages. And the parameters in black correspond to donor-related parameters, and they are fit to the DMSO data. And E-drug here in blue corresponds to the compound effect, the blocking the maturation, and it's fit to the, this parameter is fit to the on-treatment data. And if you have more, if you want to know more about the model, please come to our poster this evening. And we can discuss more. And these are the results from the model. So in gray, the experimental data is shown by the dots, and the DMSO is shown in gray, and the on-treatment is shown in blue. What you can see here is that the model can capture well the on-treatment data, and model predictions regarding the washout phase, shown in green, can be experimentally validated, as we show here. And with these results, we conclude that this model is suitable to be used to explore the dose and schedule of lenalidomide in silico, and propose regimens that could minimize neutropenia, which is a key clinical toxicity of this compound. And with that, I would like to acknowledge everyone involved in this work, in special Carla and Soraya, who developed the in vitro assay, and thank you for your attention.
Do we have questions for Cristina? Okay, so I was wondering, it's a very nice talk. I was wondering how easy it is to use to adapt your talk to compute to predict drug perturbations, so several compounds simultaneously.
Yes, we did that with other compounds as well. It's the same model can be used to fit and predict the effect of other compounds that cause blockage in neutropenia maturation, as well as cytotoxic compounds.
Okay. And also adaptation to other cancers of the immune system? Would it be require a lot of...
This assay is done in healthy donors, using cells, CD34 positive cells from healthy donors.
Okay. Okay, any other last question? Okay, so in that case, let's thank Cristina again and all the speakers of this session so far. And it's now a great pleasure to introduce our last speaker, the last keynote of the day, Georg Stelling. So let me give a brief introduction about this outstanding speaker to complete our panel of outstanding keynote speakers from today. So basically, Jörg studied biotechnology at the Technical University of Braunschweig in Germany, but he did some stays at the Kohl